Glycolysis is a central metabolic pathway in which glucose, a 6-carbon sugar, is broken down into two pyruvate molecules, each containing 3 carbons. This process occurs in the catabolism of cells and it's the first step in cellular respiration. Glycolysis doesn't require oxygen and can occur under aerobic and anaerobic conditions, making it crucial for energy production in various environments. The pathway generates a small amount of energy in form of ATP and NADH, which are essential for cellular functions and further metabolic processes. Glycolysis is a highly conserved process across different organisms, reflecting its fundamental role in biology. This process involves 10 enzyme-catalyzed reactions. Key enzymes in glycolysis include hexokinase, and this phosphorylates glucose, phosphofructokinase 1, the rate-limiting enzyme, phosphorylates fructose 6-phosphate, and pyruvate kinase, catalyzes the final step, converting phosphoenolpyruvate to pyruvate. They are called key enzymes because the reaction that is catalyzed isn't reversible, or by other means, it's catalyzed by another enzyme. These enzymes aren't available in all organs, which gives a privilege to those organs that have all enzymes to go on. Gluconeogenesis, like liver. Glycolysis consists of two main phases: energy investment phase, and this phase involves the consumption of two ATP molecules to phosphorylate glucose and its intermediate, and energy payoff phase, which generates 4 ATP and 2 NADH while converting G3P to pyruvate. Investment phase. This process traps the glucose inside the cell and destabilizes the glucose by increasing its energy and making it more reactive. Phosphoglucose isomerase converts glucose 6-phosphate into its isomer fructose 6-phosphate. This is done in order to ensure that the sugar is broken down into two equally sized molecules. Then phosphofructose kinase catalyzes the addition of a second phosphoryl group onto the fructose 6-phosphate to form fructose 1,6-bisphosphate. This further destabilizes the sugar, which prepares to be broken down in the next stage of glycolysis. An enzyme called aldolase cleaves a bond in the open chain form a fructose 1,6-bisphosphate to form two different 3-carbon molecules, one of them G3P and dihydroxyacetone phosphate. G3P lies directly on the glycolytic pathway and so go on the energy harvesting stage. On the other hand, dihydroxyacetone phosphate doesn't need to be converted into a G3P molecule before it can be used by the glycolytic pathway. Energy harvesting stage. This is the stage where all the ATP molecules are formed. The first step is the conversion of glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate by the action of the enzyme glyceraldehyde 3-phosphate dehydrogenase. The purpose of this step is to create a molecule that is more reactive and has a higher phosphoryl transfer potential. This enzyme involves the coenzyme NAD+, which is reduced. The 1,3-PGP has a higher phosphoryl transfer potential and can be used in the next step to transfer a phosphoryl group onto an ATP molecule to form an ATP. A total of 2 ATP molecules are formed in this step because 2 glyceraldehyde 3-phosphates are transformed into two 1,3-PGP molecules. In step 8, 3-phosphoglycerate is converted into its isomer 2-phosphoglycerate. In step 9, 2-phosphoglycerate loses a molecule of water, becoming phosphoenolpyruvate. Phosphoenolpyruvate is an unstable molecule, poised to lose its phosphate group in the final step of glycolysis. In step 10, phosphoenolpyruvate readily donates its phosphate group to ADP, making a second molecule of ATP. As it loses its phosphate, phosphoenolpyruvate is converted to pyruvate, the end product of glycolysis. Regulation of glycolysis. Key enzymes. Hexokinase inhibited by glucose 6-phosphate, phosphofructokinase 1 activated by AMP and fructose 2,6-bisphosphate inhibited by ATP and citrate, pyruvate kinase activated by fructose 1,6-bisphosphate inhibited by ATP, alanine, and phosphorylation. Hormonal control. Insulin activates glycolysis and glucagon inhibits glycolysis. Energy state. High ATP glycolysis slows, high AMP or ADP glycolysis accelerates. Feed-forward fructose 1,6-bisphosphate pyruvate kinase and feedback ATP inhibits phosphofructokinase 1 and pyruvate kinase. Most of the energy used by cells comes from glucose. It's the first pathway that is used to convert glucose into energy. It's the first main metabolic pathway of cellular respiration that produces energy in the form of ATP. Glucose 6-phosphate is the common intermediate that is required for various metabolic reactions like glycogen synthesis, HMP pathways, etc. Anaerobic glycolysis occurs in the absence of oxygen, during insufficiency of oxygen, and in case of the high demand of energy in the muscles. The anaerobic glycolysis pathway occurs as the RBCs lack mitochondria. They derive energy from lactic acid fermentation. Another example where anaerobic respiration takes place is in the lens of the eye.